So hello to everybody, and today I will tell you about this new ELISA test that we developed for the fluorescence Doray detection in field samples. And as you have already heard, fluorescence Doray is one of the main and the most dangerous grapevine yellow in Europe. It's a quarantine disease, and it is associated to phytoplasmas of the 16S5C and the subgroups. Its dangerousness is due to the epidemic way of spreading, and this is caused by its vector Scaphoideus citanus, which is an Ampelophagus leafhopper, which is very efficient in spreading fluorescence Doray from vine to vine. When you in vineyards grapevine yellows are present, it's very important to identify which phytoplasma strain is associated to the disease, because of two different phytoplasma strains are related different control strategies. And as Professor Bertaccini already said, different phytoplasma strains gave the same symptoms on grapevine, so we cannot rely on symptomatology to identify them. To date, grapevine yellow detection is based mainly on DNA identification of the pathogen by PCR. PCR is a sensitive and sound technique, but not within the reach of all laboratories. And it's relatively expensive and lacks suitability for large-scale monitoring. Also, serological methods are available, but they are not enough sensitive to detect the low titer of FD phytoplasmas in grapevine. Therefore, under Tropicsafe activities, a new serological detection test with improved affinity and sensitivity towards fluorescence Doray phytoplasmas was designed. This project had two main steps. First, the raising of polyclonal antibodies against the antigenic FD membrane protein IMP, and then development of an enzyme-linked immunosorbent assay, that is an ELISA, test protocol for detection of FD phytoplasmas in field samples. What is IMP? IMP is an acronym for immunodominant membrane protein. It's a protein very abundant on the surface of FD phytoplasmas. There is a high sequence variability between isolates because it is subjected to high selective pressure. And interaction with insect proteins was demonstrated, thus suggesting a role of IMP protein in vector recognition mechanisms. The slide describes the workflow of the project. Initially, primers for specific amplification of the IMP gene of the phytoplasmas of 5 group were designed. The outer parts of 2 IMP proteins, those of the FDD and FDC lambda-PM strains. The FDC lambda-PM is a variant of the FDC strains that are present in Italy. Those proteins, outer parts of proteins, were synthesized, expressed in E. coli, and then purified. And large amounts of these IMP preparations were used to immunize rabbits. And then polyclonal immunoglobulins were purified from the rabbit antisera by affinity chromatography. And then were processed into analytical reagents for application into double antibody sandwich ELISA kit. The EGGs were tested for their specificity against the IMP protein target. Western blot were carried out to assess the antisera on recombinant imp protein and also of native imp proteins extracted from reference materials like Escherichia variegatus, Vicia faba, and Catharanthus roseus. Results showed that antiserum produced against FDD imp reacted only with the strain D, recombinant or native imp proteins. And antiserum produced against FDC imp reacted only with the strain C, reactive or native imp proteins. So they confirmed the specificity and the functionality of the immunoglobulins. Given these good results, ELISA reagents were calibrated and a protocol to detect flavescence doré in field samples was developed. 58 grapevine leaves and 204 Scaphideus citanus were tested in ELISA. Together with 29 trees of Alnus glutinosa and 3 Clematis vitalba plants. And these 2 other species were chosen because it is known that they are often infected with phytoplasmas of the 5C subgroup. Most of the samples were also tested for FD positivity by real-time PCR, and strain characterization by PCR and RFP analysis was made or is still ongoing. So now let's see the results on grapevines. Here, for grapevines, ELISA tests were carried on leaf mix of healthy and infected vines. The plants were collecting in northern Italian regions, where both FDD and FDC strains are present. And in these different regions, the FDC strains are slightly different. ELISA test detected as positive 32 samples out of the 50 that were positive PCR, and detected as negative 4 out of the 5 samples that were negative also in real-time PCR. With this data, performance parameters were calculated that give a measure of the reliability of the assays. Sensitivity was 64%, specificity 80%, and accuracy 70%.
No, this one. No, not that one.
Non.
Oh, je l'ai remis là.
Il est remis.
Oui.
Ouais, t'apprends.
C'est bon.
C'est parti. Spell.
Elle, c'est Luisa.
C'est Luisa.
Mais elle, je ne l'ai pas touchée. Hein.
On l'a pas touché, hein?
Non, elle, non, je n'ai pas touché. J'ai seulement supprimé.
Can I continue?
Yes, yes. Perfect.
I see that my microphone was disabled.
There was somebody speaking, we try to catch.
Okay.
It's not you, it's our fault, or fault of somebody not current.
So only I want to say only that these 2 parameters may be improved by increasing the number of the samples, even the healthy samples. And moreover, it should be noted that the specificity of the assays toward the FD strains were confirmed in field samples, because all vines with the FDD strains were never positive to antisera against FDC, and vines with FDC strains were never positive to antisera for FDD. But if we look at the performances of the 2 ELISA assays separately, it can be seen that the sensitivities are really different, because for FDD it reaches 100%, and for FDC it was only 35%, while specificity and accuracy are similar, specificity in particular. And we don't know why. But one idea is that it can be due to the variability of the input between the 5C strains. As regards Caphoideus titanus, tests were conducted on adults collected in effective vineyards. And the results are similar to those on the total grapevine samples, being sensitivity 58%, specificity 100%, and accuracy 79%. The suitability of the ELISA test was checked also on phloem scrapings of black alder woody canes and lithium hybrids of Clematis vitalba. And all the samples were collected in Veneto region and were tested by PCR for FD positivity, and the positive samples were analyzed by RFP analysis and confirmed to be infected with the 5C phytoplasmas. Alders had showed a sensitivity, the assay on alders showed a sensitivity of 38%. So this result is similar to that found on FDC grapevines. But it should be noted that the matrix used for the test is different, and maybe from scrapings may have an effect on the sensitivity parameter, and as well as the variability of IMP protein. For Clematis, only 3 plants were tested. The results of PCR and ELISA are specular, but of course, they are only 3 plants, so we need to increase the numbers of the analyzed samples. And the last years, the ELISA tests have been made also on other plant and insect that in nature are not infected with FD phytoplasmas, but are widely used for infection and transmission trials in laboratory conditions. Those are Catarrhactus roseus, Bicefaba, and Escherichius variegatus. As you can see, results are promising, but also in this case, we need large numbers of samples to confirm this data. So we are at the end. To summarize this presentation, I can repeat that this is the first production of a polyclonal antisera against recombinant in proteins specific for FD phytoplasmas and of its use in ELISA tests. This new test proved to work for detection of FD phytoplasmas in grapevine field samples, in other plant species, both woody and herbaceous, and in insect vectors. But antisera resulted to be very specific in all the trials that were made. The test targeting FDD was more robust than that targeting FDC phytoplasma, and also for this region, optimization of detection protocol is currently underway, addressing steps like leaf conservation, homogenization methods. For example, we see that mortar and pestle are gave more reliable results than the use of an automatic homogenizer, and also on the sampling period. Anyway, these assays are suitable for monitoring programs as a preliminary screening of larger sample number. So thank you for your attention, and if you have any questions, I'm here.